Welcome to this lesson on genetic modification and the issues around it. So last lesson we looked at the techniques for genetic modification, and in this lesson we're going to be looking, exploring the ethical issues, both positive and negative, relating to the genetic modification of animals, plants, and also microorganisms. So to start with, I'd just like for you to assess your sort of preconceptions about this. So here's a kind of scale from -5 to +5. Where do you think you are on the scale in terms of your views on genetic modification? Are you strongly pro? Are you strongly against? Are you in the middle? Just write down a number. Okay, so now why? Why did you place yourself wherever you put yourself on this spectrum of opinion? What are your what kind of information and ideas do you bring to this lesson already? What are you thinking that? Just reflect on that for a second, and if you'd like, you can write it down. Okay, so in today's lesson, really we're going to be looking at a lot of different examples of this technology. You're using textbook page 231 and 232. And really what I've done is I've just looked at each example in the textbook and I've just fleshed it out a bit. So what I would suggest to you is you're just going to be making notes about the plus and negative kind of issues relating to each example, using this video, using the textbook, and you're just kind of getting an idea of the general themes behind these issues. Okay, let's jump in straight with the first example. Okay, so the first example in the book, microorganisms. So we can use microorganisms and we can modify them to make all sorts of things. One of the things is insulin. So insulin has been bacteria, E. coli, have been genetically modified. We've taken the gene, human insulin gene, this would be cDNA. I think this is done using the reverse transcriptase method, put into a plasmid, cutting it with the restriction enzymes. Plasmid goes into the bacterium. We use those antibiotic resistance markers to identify which bacteria has taken up the plasmid, and then we can grow the bacteria in a tank and produce the insulin. We can also get bacteria to produce other things to treat other conditions. Potential hazard, antibiotic resistance. If we use markers, antibiotic resistance markers, then these antibiotic resistance genes can spread through bacterial conjugation. We mentioned that just a little bit the other day. And cause increased resistance. This is a little graph here of how antibiotic resistance to various different antibiotics has spread up until sort of 2001. And actually, if we continued this graph up until 2020, it's going to keep going up and up and up and up. So, next example, plants. Okay, we can engineer plants in various ways, and one of the things we can do is we can engineer something called BT, Bacillus thuringiensis toxin, into the plants. Now, why would we want to put an insect toxin in plants? Well, the why is down to this guy. This is a cotton bollworm moth or other insect pests that feed on crop plants. And this BT toxin is toxic to these caterpillars and to other insects. It's only toxic to insects. So how do we get the gene in? Well, in this case, we use a vector which is the TI plasmid, from this bacteria we talked about, Agrobacterium tumefaciens. So basically we insert the BT gene into its plasmid, into the TI plasmid, and then we can, so this is the Bacillus thuringiensis here, and we take the toxin gene and kind of merge it into this TI plasmid, and then we can get it to go into the cotton plants. Now, it is specifically toxic to insects that feed on the plant. This is a little explanation of how. There's a toxin that basically binds to receptors in the gut of insects, and it causes perforation of the gut and death of the insect. It is non-toxic to humans because we have different cell receptors. Now, there's a lot of issues around this. Some people say it's great, some people say it's less great. And I think one of the things you have to remember is that when we're using this technology, what is the alternative technology? Well, there's two alternatives. One is a crop without any insecticide at all, an organic crop. It will have lower yields. And the other alternative is a crop that has sprayed insecticide on it. So in this case, having the insecticide in the actual crop is better than spraying a pesticide, which can kind of be blown either side of the field and can run off into water and that sort of thing. However, things like monarch butterflies might lay their eggs or feed on these tobacco plants or cotton plants, and this might cause a reduction in the monarch butterfly population. However, we always have to contrast that with what would be the alternative, in terms of using a sprayed pesticide, and it would likely have a similar effect. So, next example, soybeans. Now, soybeans are grown all over the world, both for food and for food for animals that we rear. So they're both human food and they're food for, they're ground up and used in feed for cattle, for example. And a lot of this soya that we grow today is GM. Now, one of the things that companies have done is they've made these soybeans resistant to a herbicide. Now, that seems a bit of an odd thing to do. Why use the herbicide? Well, here we have this herbicide made by the company Monsanto. Okay, it is a herbicide called Roundup. When sprayed onto two plants, if we have a regular plant which doesn't produce a specific protein, it will die. If the plant produces a protein, a specific protein engineered into the plant, then it will not die. So what this company Monsanto did was they put this special kind of neutralizing protein into soya, so that any other plant that is grown in this field can be sprayed with Roundup and all the plants will die other than the soya plants that you want to grow. So this basically makes kind of the process of eliminating weeds more efficient and increases yields. Possible risks could be if this herbicide resistance gene passes into weeds, then those weeds would obviously be resistant to the herbicide, super weeds. Doesn't appear to have happened that much to date. Here's a little graph showing the growth of genetically engineered crops in the United States up until 2019, percent of planted acres. So we can see that the soybean percentage in 1996, it was like 5, but today it's like 95% of soybeans are GM in the US. And there'd probably just be a small market for sort of something like organic soy, which would not be GM at the top there. And we're getting up into the 80s and 90s for all these things. BT cotton, the one we looked at in the previous slide, BT corn, another Bacillus Thuringiensis toxin in corn as well. And HT in this graph here represents herbicide tolerance, so like Roundup Ready. Okay, so that's modifying crops. What next? We've got golden rice. Now, this is a crop modification that I think people will be more comfortable with, maybe. So it's not a multinational corporation doing this. It is a charitable foundation doing this. So the problem is that about 500,000 children every year in India alone go blind, and some of them die through lack of vitamin A. So beta-carotene can be converted into vitamin A in the body. So this rice here, it's called golden rice on the right. It's been engineered, so we put some genes in from daffodils, so that the rice has beta carotene, so it has more vitamin A once it's eaten and broken down. This could save thousands of children a year from this vitamin A deficiency. So this is some more data on this. 2 million children under 5 years die each year around the globe because of this. So this stuff could really, really help. Now, some people were concerned that if farmers had to buy the seed every year to plant, then this would not be sustainable. So the company has offered free licenses so farmers can keep and replant rice seeds. So every year when they grow the rice, they'd sell some of it for food, but they'd keep some of the rice, which is a seed, and replant the following year, and that would still be able to produce this nutritionally kind of better rice. So similar example here for plantains, which is a type of larger, starchier banana that's a staple food in Kenya. Now, so this is a GM banana picture I found on the internet. So this is a trial here. I'm actually not sure where that is, but that's a trial. I think it was in Uganda. I think I looked at of GM bananas. So they've been grown to have an enhanced iron and pro-vitamin A contents to aid or reduce malnutrition. So plants are also being engineered to contain more zinc and also to be more resistant to pests and things. This company called Africa Harvest, or this foundation, I went to their website, so if you just Google that, Africa Harvest, here are some of the products that they are working on. So sorghum, which is another staple crop, bananas, and all sorts of things, improving both the drought and pest tolerance and also the nutritional content. Now, some people, this is a really not very valid concern, some people fear that eating genetically modified crops with foreign DNA is going to, somehow those foreign genes in the plants are going to then affect humans. Now, that is complete. Well, that is not scientifically valid at all, because of course, whatever you eat, it's got DNA in it. If you eat a carrot, if you eat some chicken, if you eat a pizza, it has DNA in it, because all food has DNA in it, basically. And we break down those DNA molecules when we eat. If I eat a piece of salmon, it does not mean that I grow gills and I start swimming around in the sea, because my stomach enzymes are going to digest those nucleotides, the DNA enzymes called nucleases and nucleotidases do that. Okay, so we talked about this a little bit. Crop plants resistant to pests. Okay, so we talked about this a little bit. This is crop plants resistant to pests. So again, we talked about Africa harvest. Now this is a sorghum plantation, very, very staple crop in Africa, not widely consumed in the UK. And this, I've forgotten the name of it now, but it's a main sorghum pest. I think it's called the sorghum leaf fly or something like that. But anyway, it's a big problem for sorghum growers. And we can try and engineer genes into sorghum that make them more resistant to pests. Now, a concern of this, and a valid concern, was that early on in the GM movement, companies like Monsanto were engineering their crops so that they wouldn't produce seeds that were viable. So that meant that every year you'd sell your sorghum, but you wouldn't be able to replant the saved seeds for the next season. You'd have to go back to Monsanto and buy more fresh seeds. This technology was called Terminator Technology, and it basically meant the science of it is a bit complicated, but it basically meant that self-fertilizing plants, when they produced the seeds, they weren't viable. So you had to buy every year from the company. And that was to protect their kind of money stream. But actually, this technology is not used widely at all anymore, because rightly there was a great debate about whether it was kind of ethical. So we don't really have this technology being widely used at all anymore. Okay, pathogens. We can engineer pathogens, and we can do this for a wide range of reasons. So one thing that we can do is we can use viral pathogens as vectors for gene therapy. And we're going to look at that in the next lesson. So here's a sort of picture of a new gene being inserted into an adenovirus. That's a flu virus. And these adenoviruses are very well adapted at getting into our cells and delivering genes into the nucleus. So we've used those as vectors in gene therapy trials. There have been some problems with this. There was a very famous death 20 years ago of a young man called Jesse Gelsinger. And here's the article if you'd like to follow that. And he was involved in a gene therapy trial. I forget for what condition, but it was an inherited condition. I think he was deficient in an enzyme, maybe in the liver. And he died, unfortunately. I'm not sure why. I think it may have been an immune reaction to the virus used to deliver the gene. And have a look at that article if you'd like to know more. Another reason that we might modify viruses to make a vaccine. So, for example, we're trying to work extremely hard right now to try and come up with a coronavirus vaccine. This would be a DNA vaccine, and it's basically going to involve taking the DNA code, or actually it's an RNA virus, taking the RNA code for the coronavirus and working with those genes, engineering those genes to try and come up with a new type of vaccine for this disease that is spreading. It's worth also saying that for this, there have been some problems with the use of viruses in gene therapy, as the allele may be inserted in the genome in a way that increases the risk of cancer or interferes with gene regulation. This is really to do with sort of more traditional, kind of old school gene therapy. With CRISPR technology, we can be a lot more targeted and direct about where the genes are incorporated into the host genome. So CRISPR technology is kind of making these concerns, as detailed over here, less of an issue. Mice. We genetically engineer millions of mice. Okay, here is a gene knockout mice. I forget which is the knockout now, but one of these mice has a gene for hair production. Well, there's many genes involved in hair production in mice, but in one of those mice, we've kind of cancelled that gene. We've taken it out. It's called the gene knockout mice. So that kind of, then we can compare the two mice and say, well, what does this gene do? So potentially it's to do with the pigmentation, because this mouse here has a sort of darker pigmentation. I think this is the original and this is the knockout. So then we can say that that gene that we've knocked out is to do with controlling the pigmentation pattern in the hair of the mice. Now, we've done this for so many genes, okay? So this is knockout mice. In 2000, there were 3,400 different knockout mice that you could buy from biomedical suppliers. Today, there's 8,000, probably close to 9,000 different types of mice now. And in total, the number of transgenic, that means genetically modified mice strains available today, is close to 14,000. 14,000 different genetically engineered mice strains that we can use in animal research. So there might be a mouse which has been engineered to be to basically have cystic fibrosis. Now that's that sounds kind of horrendous in a way, but those mice with this same genetic defect that we find in humans can then be used to investigate cystic fibrosis drugs and treatments and things like that. And in general, because mice are mammals, they have a similar cellular structure and similar organs to humans, and then we can actually study a lot of diseases through these mice models or mouse models. Pharmaceutical proteins. This is a fun one. We can basically engineer proteins into the milk of mammals. So in some proteins, mammal proteins, require mammals to need to be made in mammals so that the proteins are processed in a certain way, and we can't really get the proteins to work in the bacteria, to be produced in a bacteria. For example, the insulin one can be produced in a bacteria, but this one, alpha antitrypsin, I think it requires a bit more complicated processing. So alpha antitrypsin in normal people, it coats the lungs and the liver, and it kind of prevents white blood cells from kind of attacking them or something. We don't need to know too much detail. Have a look here if you'd like to know more. But some people have a deficiency in this protein, which means that they get lungs being broken down and the liver being broken down by circulating white blood cells, neutrophils. So this causes chronic sort of hereditary emphysema and other conditions. So we have to treat by giving excess alpha antitrypsin, this protein. At the moment, it can be harvested from blood, so donor blood. We harvest the protein out of the plasma, but we're working to try and get it into mammals. So if we engineer a cow, for example, with a human protein like this, and we put it in correctly, we can get the cows to produce these proteins in their milk. So here are some different things that cows already produce. Factor VIII, that's involved in blood clotting for hemophiliacs. Fibrinogen, also involved in blood clotting. And these other ones, lactoferrin, I think, is involved in transporting iron. So this general idea is sometimes called farming with a pH. So it's a mixture of pharmaceuticals and farming, pH farming. Right, last one. This is kind of similar to farming again. This is an example. So spider goats, okay? So here's a little cartoon of a spider goat. Now, it's not actually like that. What we're trying to do is trying to get goats to produce silk proteins, spider silk proteins in their milk, and then we can extract those proteins and spin, not webs, but fibers of spider silk. And spider silk is phenomenally strong, so we could use it for cables, for stitches, artificial ligaments, bulletproof vests, and stuff like that. There has been concern about the welfare of the goats. I suppose that's the same concern about any intensively reared animal. But these goats, these transgenic goats or cows from the previous slide, would be very valuable to companies and would likely be extremely well looked after and not be eaten because they would be wanting to try and continually extract proteins from their milk. I started to look into the mechanics of spider silk and lost quite a few minutes going down that research avenue. It's more complicated than we originally thought. It turns out spiders have loads of different types of silk spun for different things, and it's quite complicated. And also the spinnerets that kind of at the back of the spider that kind of do this to spin the silk are also important. So there's still a lot more science to be done before we can get spider goats to produce silk for us. So those are the main examples, okay? Go back over in your book, look through those, make notes on all of them. And once you've done so, move on to these questions. Okay, quite two big questions here. So these are kind of sort of mini essay questions. I'd say take the first one at least as a kind of 9 marker, and the 2 one as, you know, maybe a 5 or 6 marker. Okay, so have a look at those questions. Write a good paragraph minimum for the first one, and then the second one. Okay, pause the video and then come back, and we'll look at the mark scheme. Okay, off you go. Okay, so here you go. Here's the mark scheme. I won't read that out to you. You read through it. You see if you had similar things, similar arguments to they have, and add any extra points. And so you can, if you'd like to, you can give yourself a mark out of 9 or mark out of 6. What do you think you would have got? Could you have added extra detail? So before we go, we're just going to look at the syllabus, which is here. Now, last lesson, we already looked at these syllabus points F1 and F2. How do we genetically modify organisms? And today we were looking at this one, G. So do you feel you are now aware of the ethical issues, positive and negative, relating to the genetic manipulation of animals, plants, and microorganisms? Specifically, can you remember and talk about insect resistance in genetically modified soya? So that's BT. Genetically modified pathogens for research, so that's like viral vectors, and farming, i.e. genetically modified animals to produce pharmaceuticals. Do you understand issues relating to patenting and technology transfer? So this is, for example, making genetically modified seeds available to poor farmers and not using that Terminator technology that meant that the farmers would have to buy new seeds every year, which many farmers would not be able to do. It would be economically unsustainable. Okay. So there you go. That's a summary of everything genetic modification. And I think I'll probably also find a TED talk or something to add to this as a link at the bottom if you would like to go further. Okay, thanks very much, and I'll see you next lesson.